Okay, so problem 7.26 is kind of abstract, so it sort of makes you think about things. The system that they're using is C. elegans, the roundworm. So if you remember back to chapter 1, I think we talked about it as a model organism. They described two different genes, dumpy, I think it was, and uncoordinated, located on the same chromosome, that they recombine with the frequency, and they don't give you the frequency, right? They just say that it recombines at a frequency of big P, of P. So the first thing you have to kind of interpret is like, what does that mean? And if you remember, the frequency of recombination is what we're calculating, which then gave us the genetic distance. So that's the number of recombinants over the total. All right. So then now once you kind of understand that, right? I know it's a little abstract. It's not the best. I tend to understand things better when they're in real numbers. So as I go through, I'll kind of throw in some hypothetical real numbers too. But they're saying, okay, it's a repulsive at both of the genes, right? So that means that it's dumpy wild type and then wild type dumpy. This is one chromosome and this is one chromosome. And C. elegans can self-fertilize. So you get, the same genotype mating with the same genotype. So here you have one mating with this other one. Okay, so let's think about what are the different kind of gametes that they're going to produce. When there's no crossing over event occurring, you're going to get a DPY wild type, which you have here, and a wild type uncoordinated, which you have here. If there is a crossing over occurring, if the crossing over event does occur in the production of the gametes, you're going to get wild type, and then DPY uncoordinated, UNC, right? So you get this one and this one. So now let's think about it. When we are trying to figure out the distance between it, what do? We add each of those recombinants, right? And then put it over the total. So here's the total, here's each of the recombinants. So for each of the recombinants to equal P, right, you have to have 1/2 of this one, half of them have to come from here, and half of them have to come from there. So let's just say the distance is 10 centimorgans. So that means there's going to be 10%, right? So that means P's 10%. So 5% have to come here, 5% have to come from here. If that's the case, you can follow that through and say, okay, if P is 10%, then you have to get the frequency of each of these, you get 10%, 1 minus 10%, that's 90% divided by 2, that's 45%. And this is 45%, right? The same thing. So then you get 45%, 45%, 5%, 5%, that equals 100%. And so this, I know it's kind of mathy, right? We try not to get too mathy in this class. But so that means that each of these the frequency would be 1/2 P for both this gamete and this gamete. And now the question is, okay, what's the probability of getting an offspring that has both of these recessive conditions? So you're going to have two gametes that are two gametes that are both DPY mutant, UNC mutant, right? So you could do the same thing over here, draw that out, but it's going to equal the same, right? So that's 1/2 P here, 1/2 P here. You have to have both of those to occur. You have to have this event and that event occurs, so you times those by each other. So it's 1/2 P, or P over 1/2 times P over 1/2, or another way of saying that is P over 2 squared. And so that's how you would do that one. So it's actually pretty straightforward, but it's sometimes a little bit hard just to think about these things with respect to just sort of abstract math kind of stuff, right? So I generally am not going to ask you a question on the exam that has this. Just use, I like to use real examples with real numbers because I just think students understand that better. But here you go, that's 7.27.